thank you everybody for coming. We have a visiting faculty candidate today, Dr. Forrest Bao. Forrest is currently an assistant professor in computer engineering at the University of Akron, Ohio. He got his PhD in computer science with a minor in electrical engineering from Texas Tech University. After that, he did a postdoc at SUNY Stony Brook, where he worked on computational neuroscience in the School of Medicine. We're going to hear about some of that today. His research has been funded by the FAA, the NSF, the Air Force Research Lab, and the National Institutes of Health. He works in applied machine learning and natural language processing. I'm very excited to have him here and look forward to hearing your talk.
Okay, all right. Thank you. Thank you for the introduction. And thank you guys for coming. I know it's right before the lunch, so I guess maybe your belly is calling you. But I get it out finishing time. So today I'm actually going to talk about, since I work on quite a diverse spectrum of research, I'm going to give you aspects give you overview on my research on two aspects. One is computational neuroscience, the other is natural language processing. So before we jump into the research, I can give you a little bit of overview about, before I jump into those two topics, I can give you a little bit of overview about my research. So on my research, on one aspect, I'm very interested in the application part of computing, which mainly is on biomedical related image and signal processing. On the other hand side, which is more hardcore stuff on artificial intelligence, I'm working on natural language processing and knowledge representation. And of course, you probably look at my resume, I have a little bit of spotted publications on some other areas, which are the heritage of my undergraduate research experience. So if you ask me why do I work on so many different topics, I would like to use a quote from Carl Gauss to explain that. He said, when I have clarified and exhausted the subject, then I turn away from it in order to go into darkness again. So I think that's the cycle of my life. And if you trace back who is a PhD advisor, my PhD advisor all the way down, you'll find out that that guy is Carl Gauss. So I feel that's interesting. Okay, so so a couple of slides to show you different aspects of my work. So this is one work I did. So probably from seeing this picture, it's like this kind of a hat has many electrodes on it that captures an image, in a time series known as EEG, electroencephalograph, and it's basically time series reflecting the activities in your brain. And that work I did has been reported in my MIT Technology Review because we came up with a very efficient way to diagnose epilepsy by leveraging signal processing and machine learning. And this work, just a little bit over a year ago, has been used by a group done by MIT, from MIT and TU Denmark on using that approach I developed. And this work was reported by The Lancet, which is one of the oldest and the most prestigious medical journals in the world. And actually between these two slides, there's one thing I didn't tell you is that this work has been clinically verified by work at UCLA Seizure Disorder Center. And they published that verification work at a journal called Epilepsy, which is a top journal in epilepsy research. Something else, so this is a very complicated directed graph. You don't need to understand what that is. Actually, I don't understand what that is. But the beauty of machine learning is to teach computers something without having the domain knowledge. So this diagram shows a very complicated reactions in a cell of bacteria. And from chemical engineering perspective, they want to genetically modify those bacteria in order for them to produce certain chemicals we want. And however, in the traditional way that chemical engineers use are differential equations. But this is a very complicated reaction. So using differential equation doesn't really scale up well. So I came up with that idea. I said, since we have thousands of papers published in that area, particularly on E. coli, so many people study E. coli, and they all come up with the raw data submitted to, for example, NCBI database. So why don't we use those raw data to train the machine learning to build a regressor in order to be able to mimic this process, actually. So we did it, and the initial result turns out to be really good, better than the state-of-the-art technology that the chemical engineers have been developed. That's why we are lucky. In the end, we got an NSF fund to continue working on this, to using machine learning to understand the cell metabolism. Since I know this is the EC department, so let me show you some hardware. I also did a little bit of work on medical instrumentation. So for example, you can see here, I built a circuit, instrumentation amplifying circuit, to capture my ECG signal. And down the road, probably the natural question we want to ask the candidate is that since the president already announced the cut on federal research budget, what are you going to do? I guess the counter strategy of one of the counter strategies I'm going to use is I have quite a connection with the industry. So in the past 3 years, one of the logos you see here is either a patent co-inventor or research sponsor or paper co-author with me. For example, my NLP work is done with Amazon Lab 126. Actually, that guy is joining Google. He just signed the contract last week. And my bioinformatics work, some of my bioinformatics work is done supported by Bayer Comp Science, and some of my EG work supported by Microsoft. You can see all on my resume. So I hope down the road I can explore some connections with the industry in case the federal budget gets even worse. So before we start the main topic, any questions? All right, so the first part of my work I'm going to talk about is called human brain morphometry. So I'm going to give you explanation about what the problem is. So this is a typical image you see, probably from something you heard about called MRI, magnetic resonance imaging. So it's a typical image of the human brain. You see some black spots and white spots, and in between you probably make an imagination that this seems to be a curvy surface with many folds, right? So that's this curvy surface with folds is called a cortex, or cortical surface of our brain. Because it has folds, so it allows us to pack a large surface of large area within this structure. And only birds and mammals have this kind of folds. Other animals, they do not. And those folds are very important, actually. So how many of you know a TV show called Grey's Anatomy? Okay, actually it's a knockoff of a famous medical textbook called Grey's Anatomy. It's a real textbook. This textbook was published the first version was published over 150 years ago. So by 150 years ago, medical students started anatomy using this textbook. And if you look at the figure 726 of this very first version of the book, back 150 years ago, neurologists already started naming different folds in our brain, and they have the names for regions between those folds. So if you imagine our brain as a mountain, you can see those folds, you can consider them as valleys, and between valleys have ridges. So that's our brain. And they call those folds, they call those valleys sulcus, and they call those ridges gyrus. Okay, and they already started naming, you see they named all of them 150 years ago, so it's not new technology. And they realized by that, since then, they realized that different parts of our brain can be partitioned using those folds boundaries. For example, probably you can say on the west of Mississippi River is the west, on the east of Mississippi is the east. So they can partition our brain, something like that, with the guidance of those folds. And they realized that between different folds, the brain is responsible of different functions. So some are responsible for vision, some are responsible for speech, some are responsible for emotion, some are responsible for motor controls. So basically, to doctors, those folds are really important. And they're in particular interested in those not only the folds, but some other structures on the folds. Together, they call them anatomical landmarks. So if you imagine this is a picture of the Grand Canyon with Colorado River in the bottom. So you can imagine our brain is, first of all, we have many folds. So the folds we already said they call sulcus. And you can imagine at the bottom of the Grand Canyon you have this river. So this river is called a fundus. And if you look at this river, it has some turning points like here. So those turning points are called pit. So those are the three kinds of anatomical landmarks that neurologists defined in our brain. And they think they're very important. Can you imagine why they're important? Let's say your brain shrinks, then those structure changes, right? Indeed. So for example, here on the left is a healthy brain. It's a healthy brain. You can see the folds like this. On the right-hand side, it's a later stage of Alzheimer's. You can see the brain shrinks. The brain shrinks, and of course, those folds, their shape changes, and the river at the bottom of the folds, they naturally change because the entire fold changes. Just imagine the earthquake happened in Grand Canyon. The river will flow in a different way, right? So you can imagine Alzheimer's as an earthquake happening in the valley to help you understand. Okay, so neurologists realize that those structure changes is related with many neuropsychological disorders, such as schizophrenia, Alzheimer's disease, depression, bipolar disorders. And other than clinical practice perspective, they realize that if we can accurately locate those anatomical landmarks, we can help many problems in neuroimaging, such as automated brain labeling or registration. So to give you an idea about what registration is, so you probably heard registration in image processing a lot. But so in anatomical registration means this. So let's say we want to compare who has a bigger right hand. So let's say part of what you do is you put your right hand with my right hand, then we compare finger by finger, and we get a conclusion. who has a bigger hand. So if you want to compare people's brain, you also need to do this kind of alignment, but anatomical alignment. So let's say this is one fold of your brain, this is another fold, the same fold of my brain, that we can compare. So in the end, we can probably say who has a bigger head. But clinically, that has some other comparison we want to do, just rather than who has a bigger head, okay? Okay, all right, so this is a problem. Let's see how we can turn this problem from neuroscience into computing. So the reason this problem can be given a computational problem is, first of all, actually the raw data we have is already some data structure that a computer can organize. It's a 3D matrix, right? The 3D scan of the brain is a 3D matrix. Each element is called a voxel. And then the first thing we do is we turn those 3D voxels based on those 3D matrix. We do something called segmentation, a 3D segmentation, so we can get this vortex surface, the cortical surface. And the cortical surface, you can see, probably if you sit back, you probably couldn't see easily, but it's many, many triangles. So let me zoom in for you, a little bit for you. So you can see we use many, many small triangles to mimic the shape of the cortex of your brain. This is a technique commonly using triangle mesh, is a common technique used, for example, in mechanical engineering or in architecture or civil engineering to represent an unstructured surface. So it's a bunch of triangles. So the rest of the talk, in the rest 20 minutes, I'm going to talk about an operation just on these triangle meshes. So you can see how to represent triangle mesh. It's just a graph, right, with many, many vortex. Each vortex is one node on the triangle. With edges, the edges are the edges on the triangles themselves. And we have another dictionary which represents the 3D coordinates of each of these vortex or nodes, right? So that's the underlying structure. So if you're familiar with computer graphics, you probably know a data structure called a VTK format. It's actually our operation, we just use VTK format to represent it. And so before I show you how I did it, I can tell you what's good about my approach. So on my approach, using the algorithm with less arbitrary parameters and heuristics, so probably you realize that a lot of publications, they publish a good result. But in order to get that good result, you have to tune their algorithms to the right parameter in order to get to the right result. We don't have that. And our algorithm is very efficient. You will see down the road, actually, my algorithms are well-learned algorithms in the field of image processing or basic algorithms class you learn in programming class. So many people say, why do you use a single way to solve this problem? Your algorithm looks so easy, everybody can understand it. Well, I would like to argue that the beauty of science is to make things simple. If you can solve a complicated problem using a very simple way, why not? Why do you unnecessarily use a complicated way to do it? And of course, it's more accurate. So let me show you how we do it. So the first step is not done by me. The first step is done by an open source library known as FreeSurfer, developed by MGH in Boston. So what they do is they construct the cortical surface from a T1 weighted MRI images. So they already generate that triangle meshes for me. And I just started working on the triangle mesh. So the first step I do is I compute something called a depth map. So let me tell you what depth map is. So let's say I'm standing here, right? So let's say the valley is down there. I say I'm higher than the valley is deep. How do you establish a concept called depth? You need to have a reference point in order to establish a concept called depth. Right? Otherwise, you cannot compare who is higher, who is lower. Which point is higher, which point is lower. So how do we do it? The first thing is we establish a reference. And in that case, because our human brain is not like a valley, you can use a flat surface to establish the reference point. We actually establish a surface called a reference surface. We have something called reference surface. And by establishing that reference surface, we can measure the distance from every point on our brain to this reference surface. Okay? So then the next question you want to ask is, how do you establish this reference surface, right? So think about this. Let's say you have some food left over at home. You probably use a plastic food film to wrap it, right? So the reference surface, you can consider it as the film to wrap the food. Your food, it has high and low different points. Your food, unless it's like smooth surface, right? So how do we get this reference surface? What we did was, I can tell you what other people do. Other people probably they just construct a convex hull, which is a very common technique. But the convex hull has a problem. That, for example, let's say our brain, one of the cut surface of the brain looks like this. If you build a convex hull, then the reference surface will go from here directly to here, right? But naturally, you probably think actually this part of the brain is not really deep because nothing, they do not have any other neighbors closer to the top part. So you don't want to build a convex hull. You do not want to consider this part as deep. So what we did was, of course, let me give you an analogy first. Imagine you wrap your food with a plastic film, and you try to suck a little bit of air out of them to vacuum a little bit. That's what we did. And how do we vacuum it? We did a close operation on the original surface first. We did a 3D close operation, so that you can see you can bridge those small structures. And then, of course, when you do the closing operation, you can have different constructing elements of different diameters. How would you say that? Radii, right? So we try different radii, and in the end, we pick one of the reference surface such that the sum of two parameters are minimized. First is the surface area, which means it does not expand too much, because if you suck air from the food film, it will expand too much. Second of all is we make sure that this reference surface, the nearest point on the original surface, the reference surface, the total sum of all of them is being minimized. So we minimize the sum of these two parameters. So which means we try to let the food wrapping film to sink a little bit toward the food, but it does not expand too much. So that's the goal of it. So in the end, we can get the optimized reference surface. And to tell you how do we measure, when we say it's called travel depth, it means that, so for example, this point and this point, if you want to measure the distance in a 3D space, the easiest definition of distance is Euclidean distance. But that may not make sense for our human brain, because human brain may have a structure like this. It's a wrap, right? So you don't want the shortest distance to cut through this surface. You want to have some anatomical meaning, let's say. So what we did was called something called travel distance. Travel distance is the shortest distance between 2 points in 3D space without going through any of the surface. So which means the travel distance from this point to this point is actually represented by this path. So that's what we mean by the travel distance. So in the end, by first of all establishing the reference surface using closing operation, finding the optimal radius for the constructing element, and then measure the travel distance, we can generate a travel distance map on our human brain, So you can see the blue part, the bluer it is in this color map means the shallower it is. You can see the ridges, they are all very shallow. They are in color blue. And you can see between those ridges, we have lighter colors, which represents the depths. Okay, that's the human brain, the depth map we established. So, to show you the result. So in order to get the sulcus, the sulci, which is the plural form of sulcus, which is all the folds in our brain, we can do a very simple thing. We just do a thresholding on the depths. We took the median of the depths on the entire hemisphere as the threshold. Anything above it is the ridge. Anything below it is part of the fold. So we can get all the folds. You can see it's all the folds we have. And the deeper you go, the color goes redder, right? So that means we really established a meaningful depth on our human brain. All right, so the next thing is we extract those pits, which are the bumpy points on our brain, and we define those pits as local deepest points in our brain. So you can see if you want to find a local minimum, it's very easy, just do derivative within a neighborhood, right? For every node on the graph, you check whether it's the local deepest points around its neighborhood. But of course, that's going to take a really long time to check, because for every of them, you have to do repetitive thing. And actually, you do something repetitive. For example, let's say, here, as you have one node here, and has this node A has two neighbors BC. So you compare A and B. You want to check whether A is local minimum. Actually, later, if you want to check B, you probably go back to check A again. But since you already did the comparison, there's no need to repeat it. Of course, you can store the result of this comparison. So we did a way to make it even easier. We just sort all the vortices based on their depths. And then we just check from the deepest points all the way up to check, make sure all the nodes, their neighbors, has not been checked before them. Then we know that they are the local deepest points. So this algorithm, you probably heard about it, it's called a flooding algorithm. It basically simulates the approach that water falls into a structure. So any node, for example this node, let's say this node, this solid blue node, its neighbor is already below the waterline, so we know that this node is not a local deepest point. So we skip it, and so then in this way we can find all the local deepest points. And another important information we get from this flooding algorithm is that, for example here, we can see that these pond and these pond, they got interconnected through this node. Because if water level increases to the level of this node, we know that these two ponds are going to merge together. So which means in this way we can also know that how our human brain branches into different depths. Because later when you want to compare two structures, you can use how they're branched to compare them. Okay, so that's how we get the pits. So this is the visualization of the result of the pits. You can see they are kind of like sugar fostered on some kind of food. As you can see, they distribute mainly at the deepest points, at the bottom of each of these valleys in our human brain. And you may say it's hard to see. Don't worry, I'm going to give you an inflated brain to help you visualize it later. And so we already got the structure, the fold, and got the pits. The next thing we're going to get is the river at the bottom of the valley. It's called fundi. The reason we do it in the end is because it's kind of difficult to do it. So first of all, I'll show you the result. The result looks like this. So I made each of the folds a little bit semi-transparent. And the blue curves you see here, the blue curves are the river at the bottom of each fold. Looks like making sense, right? Like here, 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 here. Looks like making sense. So how did we do it? I was thinking how to do it. Many people try to do it in different ways. And I was thinking about one fundamental algorithm we learned in algorithm class called minimum spanning tree. How many of you here know it? Okay, pretty much everybody knows that. Good. So in some other EC department, I have to explain what a minimum spanning tree is. So I was thinking about this. Let's think about how a river forms. Let's say yes, it rains now. Let's talking about a surface. Let's say this is a road. This is a surface. It's totally flat. If it rains, what's going to happen? Then the water is going to have same depth in every point on this ideally flat surface, right? So, but if it has certain fold like this structure, then water will mainly accumulate into this part because it's deep, and the gravity will drive water to accumulate here. So I was thinking, what is river actually? If you think about the depths and you invert, if you build a graph whose edge weights are the inverse of the depths, then the river is actually the minimum spanning tree. Because the minimum spanning tree is what? The minimum spanning tree is a tree that has the lowest cost in this graph, that connecting all the vortexes, right? So if the edge weights of this graph is the inverse of the depths, then this minimum spanning tree actually is a tree that maximizes the depths without creating a circle or a cycle, but it visits all the vortexes. So I'm thinking that actually the river is what? The river is actually the main branch of this minimum spanning tree, and the minimum spanning trees themselves is the path that rivers goes on this graph to go into this river. That's the minimum spanning tree looks like. I mean, that's how we can use minimum spanning tree to computationally find this river. Everybody else follows that idea? Okay. So if you, well, it's partly hard to explain this. It's very easy to explain in West Texas because West Texas, it's very dry, but when it rains, we don't have much plants on the ground. So you can see many, many valleys. They have many, many curves on the side. Those curves are part of those branches of the minimum spanning tree. Just imagine water flows through them, so that's why the river, the valley, forms into that shape, because water flows and then takes the soil away. Okay, so the river is, remember what the river is. The river is actually a path connecting the deepest points in the bottom of the valley, right? And so a river system, which includes the mainstream and the tributaries, they must be the minimum spanning tree of the inverse of the depths, or the negative of the depths, by the definition of the minimum spanning tree. So in order to get the minimum span, in order to get the mainstream, all we need to do is to pruning the tributaries on the minimum spanning tree. Get rid of the shallow branches, only leave the deepest branch, which is will be the mainstream of the river. So what we do is we're doing the 3 steps. The first thing is we want to decimate the mesh. Imagine why. Because building minimum spanning tree is very costly. The time complexity with number of vortexes and edges you have. So we want to decimate the mesh to make the algorithms run a little bit fast, but this may not be the problem down the road. And then we build a minimum spanning tree with respect to the negative depths of all the vortexes. And then we prune the minimum spanning tree to get the mainstream. Of course, we need the guidance of the pits because there are local deepest points. Okay, any questions? Okay. All right, so we first decimate the mesh to tell you why. So in one hemisphere, this is a number from one hemisphere from one subject spring. It has this many nodes, this many triangles, so it's going to take very long to run. So in order to decimate, what I did was just randomly pick vortexes, remove them, and all the edges connecting them with the neighbors. Of course, when we do this, we make sure we do not break the connectivity of the graph. We do not turn a graph into two separate graphs. And then we just generate the minimum spanning tree, which is a standard problem in data structure. So you can see the result. This is 3D projection, of course. So you can see that what happened was we have certain branches at the starting from the very deep part of the valley. And you can see many small branches. They basically like leaf nodes. They end up on the shallow parts, like here. And they all go from the bottom to top. They fall and end up in the leaf nodes at the shallowest points on this graph. So it looks like it's making sense, right? But let's see closer. So let's imagine that you can inflate your brain like a balloon. So it's like this. So the path has been color-coded. The redder it is, the deeper it is. The greener it is, the shallower it is. So you can see we have the main branches, and we have many, many small branches. They go to shallower places, right? Okay, so all we need to do is to get rid of these shallow branches, then we can get the main branch, which is the river. So if we zoom a little bit, you can see it's like this. So we have many, many small branches on the side. Just get rid of them. So how do we get rid of them? Let's imagine you have a tree in your house, in your backyard. Now you're moving your house. You want to take the tree with you. What do? Let's say the tree is really, really tall. Let's say twice of my height. You wouldn't take the whole tree, right? Because there are going to be too many branches and too many leaves. In the summer, the tree may die very easily, right? So you want to get rid of some branches. So what kind of branches do you get rid of? Do you get rid of the main trunk? Probably not, because you're going to kill the tree, right? So you get rid of small branches. So what we did was, we first of all sort all the leaf nodes, because for each branch, actually, we know their depths, right? So we just start from, we can sort them, and we start from the shallowest. Let's say this one is the shallowest, we get rid of it. Then this whole structure becomes another branch, right? So if it happens to be the second shallowest, we get rid of the whole thing. So we gradually remove the branches until we reach a branch point. So that's what we'll do. So we start from the leaf node, from the shallowest leaf node. Then we remove the immediate edge connecting this leaf node. So it will be the edge AE. We remove it. So then E becomes a leaf node, right? So if E happens to be the second, E becomes the deepest shallowest leaf node right now. So we remove its immediate edge, which is EC. We remove this edge. So now, because remember C is a branching point, so we stop. This entire branch will be, the pruning will stop at point C. So when we will start another branch, we go continue pruning it in this same process until we hit a pit, which is point D. So then we stop, because we don't want to get rid of local deepest points. So in the end, we can get this result, which we already see a few minutes ago. Let me show you, if we inflate our brain, it looks like these. So each of this color patch is one fold in your brain, and we build one minimum spanning tree in each of these folds. And so this fold is color-coded. The greener it is, the shallower it is, the redder it is, the deeper it is. And then we have this blue curve. Each of these blue curves is one river we generate at the bottom of the folds. So you can see, looks like they make sense, right? Looks like they form some kind of river, like this. Looks making sense, right? Okay, so can we just be happy and have a happy ending at this point? So if you are a reviewer, you want to kill my paper, what would you do?
Evaluate.
Yeah, so evaluate. We can't just visually evaluate it, right? We want to see how it really looks like compared with the ground truth. So where does the ground truth come from? Remember earlier I said a neurologist, they partition our brain into different parts based on those folds, right? So let's see how our folds, our rivers can partition the brain compared with the neurologist's partition to the brain. So this is one result. So each of these color patches is one region labeled by well-trained neurologists. So you can see neurologists say, this is one part of the brain, this is another part of the brain, and naturally we have the boundary between these two parts. So we can compare the river we extracted with the boundaries formulated by the neurologists, and we can compare their distance. So there are many ways to compare the distance between two 3D curves. For example, one of them is called a Hausdorff distance. So one way, so in order to save time, I didn't show you the tables of the result because many, many tables. You can see, I can jump back here. You can see the numerical results in our recently published papers. There are three approaches on the planet right now. I can extract those rivers. One is us. and then there are 2 other approaches they published as well. And our approach, the accuracy is in the middle between these 2, but our algorithm is, it's more cost effective than the other 2, and you can determine how long our algorithm is going to run. It's bounded. Some other algorithms, for example, they use Monte Carlo iterations to gradually find those rivers. So the question is, where do you stop? You can't guarantee that we'll stop at a certain point. But think about minimum spanning tree or sorting, they're all bounded algorithms. We know their boundaries. So I would argue that we can get the middle accuracy with the best simplicity. I would argue with that. Okay. So can we just have a happy ending here, knowing they're accurate? Is that good enough? We want to go down the way further, because just knowing that we can partition the brain accurately is not enough to help the diagnosis tasks. So what we need to do is, we need to turn those 3D structures, for example, turn these fold into a bunch of feature vectors. The reason we want to turn them into feature vectors is because we want to throw these feature vectors into machine learning like a classifier. So we can say that, okay, this fold is from a healthy subjects, or it's from a patient at early stage of Alzheimer's. So in this way, we can diagnose we can diagnose Alzheimer's much earlier by checking the shape of this fold. So the question is, how do we turn a 3D structure